Okay, well, welcome everybody. And today we're going to have some really interesting talks, as always. Data quality considerations for evaluating COVID-19 treatments using N3C, principles for accommodating missingness, and role in analytic decision frameworks for EHR-based studies, and the MLP team update past clustering pipeline and GAP insights. And so moving forward in the next few weeks, we have the Rare Diseases and Rare Disease Network talk that I've been actually very curious about. This has been scheduled for a while, along with the new N3C Public Health Dashboard demo, April 18th, semantic clustering, and the monthly Recover long COVID team updates with the speakers listed to the right. I guess we still have April 25th not completely nailed down. May 2nd, practical machine learning for COVID-19 using EHR data case study, and then the BARDA challenge updates, which I'm actually very curious to see how that's going. As another dubious distinction, we're almost up to 5 million COVID positive cases, which is remarkable. I remember when it was a few 100,000. So hopefully things are calming down a little. Okay, so Hythem Sidky, are you going to be going first?
I guess so, yeah.
Your slides are right here, so we'll look forward to your talk. And so I think you're going to share your screen, right?
Yes, correct. All right. All right, can you all see my screen?
Mm-hmm.
All right, let's hope this. Awesome. All right, well, thank you all for being here. I'm Hythem Sidky. I am a senior data scientist at ITRB at NCATS, and I wanted to talk about data quality considerations for evaluating COVID-19 treatments using N3C. It's a bit of a mouthful, but this is especially in the context of GAP, which Dave Sonner leads. And this is some of the findings that myself and a bunch of wonderful collaborators have put together as a summary of looking into treatment effectiveness within the context of inpatient drug therapies for COVID-19. And some of this stuff that I'm going to be talking about, maybe some or all of you have heard about and are familiar with, which is fantastic. And if you're not, then even better. But the basic idea is, so to give some context, as we just saw from Joel, N3C has a lot of data. We're covering about 13 million patients, 72 data partners, and growing. And one important thing to keep in mind is that these data were not created for research. They were created for clinical and billing purposes. And what that means is that if we want to responsibly use these data for research, we have to understand, number one, the complexities in these data, and of course, specialized study design and methodology, depending on what you're trying to achieve. So what I'm going to be focused on here are complexities in the data. And again, just for context, we're looking at this in the context of defining a patient population using criteria that are typically used in observational inpatient drug effectiveness studies. And the things that, the findings that we're going to be talking about are not specifically for any particular study, but I think they apply across the board, especially in the realm of translational sciences, where folks are applying machine learning and all kinds of other techniques to get at the same very important questions. And especially if we're thinking in the context of maximizing causal inference, then these things are going to be very important. Now, the first category relates to data missingness, and I'm not going to go into a lot of detail here, especially since Ken and others are going to be talking about dedicating a talk to this. But I just wanted to highlight a few things. So, as a very first step, one thing that's important to do is to identify the variables of interest for your particular study that are going to be meaningful. And having access to EHR data means that measurements, so things like respiration rate and heart rate, O2 sat, body temperature, and other labs are very important. And that means that it's very important to filter out, potentially, the data partners which don't have sufficient data completeness on the variables of interest. So what we did here is we used hierarchical clustering to basically group together all the sites that satisfy our particular requirements. And this varies again from study to study and per the researcher's requirement. But as think of this as me walking through like a flow chart, like a flow diagram of the different things that we would accomplish in order to essentially end up with the right inclusion and or exclusion criteria for a specific study. Now, kind of concomitantly with this hierarchical clustering, it's important to also consider temporal patterns along with patterns that occur at data partners. So just to kind of go back here, the level we're talking at here is completeness at the level of data partner, but there might be a data partner that meets whatever threshold that you set. Maybe you have like a certain number of standard deviations above which you might filter out a data partner. But then one thing that's interesting is if here we have an additional, like some lab measurements like white blood cell count, bilirubin, creatinine. And what's interesting is some data partners do have missingness all across the board. So the fraction of missingness is very close to or equal to 1. But in other cases, you see interesting things. You see temporal patterns. So for example, here for this data partner, the fraction of missingness increases over time. And all of a sudden it appears to go to 0, but in reality, we're getting very little data from them. And then other data partners have very similar patterns, and it really varies. And my goal here isn't to get into how you deal with this. I'll leave that to the next presentation, and they can talk a little bit more about how you handle missingness that's not at random. But my point is just draw attention to the fact that sometimes you might look at completeness overall as being a threshold for including certain data partners. But in fact, when you look at things as a function of time or geography or any or other variables, you actually see heterogeneity. And that's something that's very important to factor into your analysis and into your inclusion or exclusion criteria at the level of the data partner. Now, I do want to just put a shout out to the Logic Liaisons team, which have already compiled and released templates for diagnostics for exactly these kinds of things, right? So when we started this, those didn't exist, but there are code templates right now that are out there, and I think more, in the works that allow, folks to go into the Nod store and pull together templates and use them to look at things like data missingness and completeness for specific key variables of interest for their specific study as a function of data partner and time and things like that. Now, moving on from there, we have drug coding specificity. And so one of the challenges here is that, drugs are frequently coded at the ingredient level. What that means is oftentimes you might look up an RX norm ingredient and you get all the descendants and pull all specific or pull all the drug exposures that relate to that RX norm ingredient. The problem with that, so here I have dexamethasone as an example. So the problem with that is when you have something that's coded at the ingredient level and nothing else, you actually don't have information on the dose, strength, route of administration, other important information that might affect the kind of analysis that you are interested in. In particular, we all know, that certain drug therapies, the dosage and the route of administration has an impact on the effect of that specific treatment on a patient. And therefore, that's something that we should factor into our analysis, whether that specific therapy is the primary treatment that we're trying to study the effect of, or it's something secondary we're trying to adjust for. And in that case, the researcher has to make a trade-off between how sensitive they want to be and how specific they want to be, depending on the application and the role, the function of the specific drug that they're looking at. Now, going along with the idea of drug therapies is drug exposure duration. So, folks have spent a lot of time looking through OMOP. They know that there's a drug exposures table and there's a drug errors table. Now, the problem with the drug errors table is that it was designed, it's a derived table. And what that means is it's basically, there are a set of rules that govern its construction from the drug exposure table. And one of those rules is that any drug exposures that happen within a 30-day interval are merged together as though they are one era, so one long exposure. Now, that might make sense for chronic, outpatient medications that a patient might be taking, but what if you're interested in critically ill patients, short-term acute drug therapies, acute treatments? In that case, those criteria are not actually suitable, and you have to come up with your own drug era definition. Now, it turns out when you start looking into things, the data reporting from the data partners varies wildly. What that means is sometimes you might get open intervals from a particular data partner. So how do you handle a case where a patient has a drug exposure start date of 2/27 and then no drug exposure end date? So it turns out, and the point isn't to kind of get into the weeds, but just at a high level, it turns out that there are like 4 or 5 dominant patterns that you actually observe amongst the different data partners. And even when you come up with logic to reconcile all of them and to merge them into your own drug errors table, you're still left with many open intervals. And you can test a number of assumptions of like, how do you handle an open interval? Do you assume that an open end date means that a patient is still ongoing treatment up until the last data pull? Or does it mean that the exposure is a single day exposure and it ends with the last recorded date? So with something like dexamethasone or a steroid, it's kind of hard to get to the bottom of it. But something like remdesivir, which has a very well-defined, course of treatment, that would be either 5-day or 10-day, et cetera, with increasing days from there on out, you can actually start testing how certain hypotheses or certain assumptions affect the overall distribution of treatment duration. So on the bottom left-hand side here, you can see that the remdesivir treatment duration for patients, we've excluded, for example, treatments where the exposure ends with a patient dying or a patient getting discharged, and a number of other edge cases that might essentially confound or pollute this kind of distribution. And what we find is, while there are a large number of patients who do have a 5-day treatment duration, there's even larger number of patients that just have a 1-day exposure. And it's fairly unlikely that remdesivir exposure to this many patients was just for 1 day. Granted, there could be adverse reactions, et cetera, all these other things. But when you dig into these data, you find that these 1-day exposures are dominated by, so there's an additional field in the OMOP tables that indicate the source of these data, and they mention that these come from claims, but in reality, they might be billing artifacts or essentially data that's extracted that does not come from the primary EHR that data partner might be running. So the upshot of this ultimately is that in some situations, you have limited ability to get full time resolution on that duration of exposure for a specific drug, which matters a lot when we're talking about the exposure on the order of days for critically ill patients. And ultimately what that does is it actually limits the kinds of modeling that you can do. So if you're interested in looking at time varying effects and doing more sophisticated longitudinal modeling, you're actually going to be limited based on the availability of drug exposure intervals from the different data partners. Now, on the clinical outcome side, one very common thing that we see in studies looking at treatments for COVID-19 are composite outcomes. So, for example, you might have, in-hospital mortality or discharge to hospice. But then, unfortunately, discharge disposition is actually frequently missing. As you can see here, overwhelming majority of cases, at least in our investigation, were null or no matching concept, which are kind of the same thing. And discharge to hospice kind of shows up down low here. And one thing that I learned, I don't know if it's still the same because these things are changing, day to day, is that CORNET and OMOP are the only 2 CDMs that support discharge disposition. So again, that might change in the future, but when we looked into it, that was the situation. So, discharge to hospice, this about 1,800 patients in our case here, it becomes very difficult to justify that composite outcome because we don't necessarily have very good capture of hospice. Another thing, of course, is mortality as an outcome of interest. And, if you look at mortality, prior to, again, and this really shows how things are changing all the time and it's very dynamic, but, one challenge with mortality and EHR data is that those typically represent in-hospital mortality, right? So if a patient is discharged to hospice or something else happens and they die within the specific follow-up period of interest, we don't necessarily know that, which means we have we could have a pretty substantial underreporting of mortality. Now, PPRL linkage does address that problem, but one other thing to consider is, when we're looking at mortality, even from within the EHR, within a specific visit, this is just a distribution of the difference between the death date of a particular patient and the end date of a particular visit. we've filtered out a number of edge cases that we can think of. And if you spent enough time talking to some folks, you've heard of zombie patients, and we've eliminated those as well. And even after doing all that, you find some patients that die well before their visit end date. And you find some patients, and this is quite long-tailed actually, this is somewhat concerning, a large, patients where the recorded death date is significantly, after the visit end date, and they don't have any visits recorded after that either. So, there could be interesting things going on there. We also noticed in some that oftentimes the date time that we receive from the data partner only has the date, so we don't get timestamps. And what that means is that if a patient actually, the death is recorded on a specific date, maybe late in the day, and then, the physician only signs the encounter and, closes that visit, like 12:01 a.m. the next day, you might actually have a difference of plus or minus a day, depending on when that happens. And that has to factor into what one defines as in-hospital mortality in that situation as well. Another scenario we found is that some data partners, I think there was actually only one, they did not shift. It seems like they unshifted their data. So when you look at the level 3 data, the LDS, you have unshifted or non-shifted data, but then it seems that the death date was still shifted, and there was a uniform offset across all their patients. So it is very important to look into all these different nuances of the data. Another outcome, for example, could be an acute inpatient event, whether it's DVT or PE or an MI, stroke, pulmonary edema, et cetera. In that case, you have to look at the reporting pattern of a specific data partner. So some data partners, for example, they might record prior medical history daily during a patient's visit, which essentially eliminates the ability to differentiate between a new and a recurring event. So, for example, a patient might come in with a medical history of MI, and then because that's on their medical record and their medical history, that shows up with a timestamp for that specific day during their entire visit. And that doesn't mean they're having MIs every single day. It just means that we can't resolve whether or not that is the record of MI is a consequence of that data reporting pattern, or that patient may have actually also had an MI during their hospitalization, and we can't distinguish between them. Another very, very important aspect is evaluating baseline medical history. And now, what does that mean? That means that in a modeling task, we typically want to adjust for specific comorbidities. So pre-existing comorbidities. One very common thing to look at is the Charlson comorbidity score, or maybe other conditions a patient might have that are not part of the CCI. And in that case, we're dealing with EHR data, and what that means is there's no enrollment period. So this, unlike claims, there's no specific enrollment period, over which we know very, very clearly that if something is recorded for a patient, they had it during that period, or if it's not, they did not. So essentially, the absence of a diagnosis of, let's say, diabetes for a patient doesn't mean that they don't have diabetes. It just means that data wasn't recorded by that specific data partner that transmitted that information to us. Now, there are different ways of characterizing EHR continuity, and this is just one of them. So if you have a different way of looking at this, that's perfectly fine. But here what we have is the distribution of months of prior history. And very specifically, what we're looking at is, what is the earliest date on record that we have a visit for a specific patient? All right, so the earliest visit information, the earliest visit date that we have for a specific patient with a given data partner. And what you can see here is very interesting. There's a large number of patients that have no months of prior history. So their first visit, the first recorded visit with a data partner is their admission, their COVID-19 related admission. And then we have this, a number of patients that, let's say, moderately uniformly distributed between 1 and 23 months. And then we have another large peak at 24. Now that large peak at 24 simply means that it's really 24 months plus. And that's because of how the prior history is structured in the enclave, which is that data are transmitted as early, going back to January of 2018. And what that means is a patient who's admitted for COVID in 2020, let's say early 2020, we have 2 years of history on them. But if a patient is admitted in 2021 or 2022, we would have more history going back to 2018. So in order to normalize that effect, we're looking at 24 months plus. So what we can do is we can say, well, let's look at how these different populations differ. So we have the no history group, we have the 1 to 23 month history, and the 24+ month history group. And as you would expect, so the no history group patients are younger, they're more female. They do have a different distribution of race and ethnicity and BMI. But what I want to point out here is that across all essentially chronic comorbidities that we looked at, the prevalence is lower for the no history group. So they have less recorded hypertension, diabetes, CAD, CHF, COPD, et cetera. And that might seem to be intuitive to some folks, but what's interesting is they have a larger percentage of patients who are ventilated on admission. So the severity of the cases is higher for the admitted patients, and in hospital mortality is higher as well. Now, what's interesting to point out here, of course, is that rather than specifically conclude something about the group of patients who have no history, what we can say is we actually have no ability to differentiate between the different between the makeup of this particular group of patients with no history. Because as I'm sure we're all thinking about, these could represent patients, number one, who are perfectly healthy prior to their hospitalization. That's entirely possible. It could also represent patients who have very poor access to healthcare. So they are patients with these comorbidities, but because they have worse access to healthcare, they don't get recorded appropriately. Or they could genuinely be patients who were transferred to, let's say, a regional healthcare center, which make up, I think, a large number of our data partners. And because they were actually seeking care elsewhere, we're not able to actually look and see all of their history. So it could be and probably is a combination of all of the above. And it's not necessarily straightforward or even possible to tease out, which is which when you're looking at patients with no history. And what that means, it's very important to factor that into account when you are doing your analysis and your study. This dives in a little bit more into the EHR continuity. And here we're just looking at the standardized mean differences between patients with no prior history and with some prior history. And what we can see is that the largest differences are essentially cardiovascular disease related, either treatments like statin use or ACE inhibitors, or actual chronic conditions like hypertension as well. And once again, just to really highlight that, looking at things multidimensionally, like looking at joint distributions is very important. Here we have essentially the fraction of patients with no history as a function of data partner, but also as a function of pandemic month. So that will give us insight into whether or not the reporting practices or the nature of the patients or all kinds of things vary as a function of time. Is it because we have less reporting from certain data partners because they were overwhelmed and medical history wasn't being pulled over? Maybe care everywhere linkages weren't there. There are all kinds of possibilities, and it's also this is why it's very important to look at things as a function of time, in addition to looking at sort of univariate factors as well. And so with that, I think I'll end here. And yeah, thank you for listening. And I hope this was beneficial.
Well, thanks. Well, that's a really interesting dissection. So let me start. And it seems to me that this could be really the basis for a methodology chapter in a book. I guess we are doing a book or a paper, because this is obviously very generalizable with lots of implications that in a short talk, I'm sure, most of us certainly, I know there are lots of implications. It's tough to really grasp them all, but certainly, one could imagine writing a line like with statistics sort of book, and you could generate all sorts of crazy hypotheses and then construct composite populations that happen to have the differences in disease incidence, prevalence, this, that, and the other, and come up with all sorts of crazy conclusions, really just to demonstrate that you could do it. So do you have any thoughts about generalizability of this stuff?
Yeah, so we do have I absolutely agree with you on the generalizability. I think these principles apply across the board. And of course, there are some, when it comes to decision making, that varies considerably depending on the kind of study you're doing and what you're interested in and the kind of analysis that you're performing. But this is something, so we've actually, we've written a paper and we're about to submit it imminently. So I think it should be available very soon for folks to go ahead and read, but we are ultimately interested in, so this is one half of sort of the coin, and this is on the data, the data quality side, but there really is a study design and methodology side that goes along with it, because again, the decisions that one makes based on these kinds of observations varies based on the kind of analysis they're doing. So we have that in the works as well. And the final piece is, this is a part of also the broader, GAP initiative. So we plan on contributing these kinds of things to, GAP moving forward. In addition to, myself working on contributing some templates, like diagnostic templates, to Logic Liaison so that they're available to everybody. So the hope is to make this available to as many folks as possible so they can go through and be able to diagnose and think about these kinds of problems. We can't give everybody all the answers to all their problems, but we can at least make sure they think about these things before they go ahead and continue with their analysis, if they haven't thought about them already.
Yeah, great. Other questions, comments? Okay, great. Well, thanks. Okay, so the next talk is, I think, assuming it's in the correct order, principles for accommodating missingness and their role in analytic decision frameworks, EHR-based studies, Ken Wilkins and Harold Lehman. So, okay, great.
And Elena Casiraghi, who's also on the line.
And Elena Casiraghi. Apologies.
I think Hythem's done a great job of setting the tone here, and our subtopic here and looking at accommodating missingness and the role in analytic decision frameworks. I got a lot of material on it. These slides can be made available after the fact in terms of the principles, but I'll cover these first two bullets, and then Elena and Harold can pick the last two. Quickly to move through, you can see that I have some, not just that outline, but also how to find out more with some links for those that get the slides after the fact. But this is really a context of, you have intended information in your N3C research that's not complete, as Hythem has already nicely outlined. And we want to be able to point out to all the researchers involved in N3C that, there are N3C-wide teams that can help you with this, from data liaisons, logic liaisons, to the machine learning pipelines that we'll hear about next after this subtopic, to folks like the Applicable Data Methods and Standards team and Palantir teams that can kind of help you work through when you're faced with incomplete data relative to what you ideally would have, and had you design your own study and capture your own data, what you can do with that. So really, we all know about how, despite having this across the nation availability of data partners and healthcare systems, all the information from the clinical encounter, even the community context of an individual patient who's contributed their data through to N3C, it all gets winnowed down to what you get down in the end, as pointed out by Hythem. And so we as researchers inherit this sort of above info loss, but there's got to be principled ways to inform how we mitigate the info loss to the extent possible. And it really, for us, it goes back to what's done centrally within N3C, and that also can inform what happens with certain data partners in healthcare systems. And it does set up this framework that each of these sort of points at which you brought down, there's actually some published literature on how to kind of break out those different mechanisms and how incompleteness of information is impacted all the way through. And you might actually allow different modeling approaches within each of those steps. So we already know, that it's an unprecedented resource here, the National COVID Cohort Collaborative at N3C. And the same, one of the prominent papers I was mentioning here is it says the importance of addressing missing data will only increase as the amount and the corresponding complexity of EHR data increases. And you could see that just from the diagram that we've seen in other N3C talks about how across different common data models, all this harmonization is doing to bring it to the OMOP common mapping. And from there, you still, even within what you get, because these data are not, they're there for clinical care, they're there for billing and other purposes within the healthcare systems, they might have recorded values without even putting any unit in there. So you can see what already has been done by the multidisciplinary teams of N3C, where they inferred missing units and filled in a great amount of it. And beyond that, even past that point of data ingestion harmonization, with the work that the phenotyping and data acquisition teams have done in that work stream, the logic liaisons have pointed out that there's still some systematic missingness here, as you saw last week in our GAP initiative talk that involved logic liaisons across a large number of variables. So, the idea is that you taken this into account when choosing the current snapshot of data. There are certain data partners will improve what they have later on, but whatever you have currently for your given question, you can filter on that. And that's the idea of using observable operational feature, like some sort of metadata that it's going to help you import the right amount of information that's reliable from those sites, as Hythem's pointed out. And you can revisit that talk from last week, or as I show here, you can see that there's issues that could be done as already covered with Hythem's talk, where you can look at over time as well as within data partners within specific types of variables. But the idea is, like, you might be asking yourself, okay, I've always heard about this missing data. It's kind of a hobgoblin of people who deal with missing data methods, right? Why do you need to worry about it? You can just drop observations with missing values, right? I can just impute the missing values. Well, that's gets into how you impute. What is the model for that? And that's what Elena's going to look at next. But ideally, you shouldn't just cross your fingers and hope for the best. That's not necessarily a defensible scientific approach, right? So, but with some principles in mind, we can do some principled planning, and you can go beyond the very restrictive assumption that involves dropping observations where you think, that's why would you not do that? It's akin to thinking that those with missing values are a completely random sample of all. And we can already see, if you look empirically at the data in N3C, that conditioning on some observables and getting some prediction of the probability of getting things missing, you can see that there are very significant, strongly predictive factors that already show that that's not the case. So you would try to work your way toward a more relaxed missing at random assumption that we'll get into in a second here. But how to go about and adopt a specific missing at random assumption gets into how you would impute the missings giving the observables. And I'm trying to avoid some equations. You may see some later, but try to just keep it at a conceptual level. In the end, you'll go from this green thing where you're spot on your target to something off target. And there's still this expectation in peer review that you'll have dealt with your missing data, as you see here, and start, if you go to the structured template and reporting tool for real world evidence, as adopted in a lot of regulatory contexts, or record the STROBE, or strengthening reporting of observational epidemiology extension for EHR-based data. It's all part of those things in there. So to have some idea up front of how to consider the way the data got to you and how you're actually making choices to get your way towards some findings from a given analysis, it's all part of what's expected in the peer review. So that's the what. I'll round out the picture of the what and demystify a bit of what these data fixtures and assumptions really involve. And later we can get into the how, the pragmatic resources you can have at N3C from Elena and from Harold, as well as other groups that are not listed here, part of the GAP, the Good Algorithm Practice Initiative team of teams. So I had an example here, but I think in the interest of time, what I might do is to say, it's even going to apply when you're trying to do just a strict predictive model, right? You're going to end up with predictions that may be off. And so the first principle is decide your primary target estimand in its ideal form. This example was just trying to look at one variable amongst many that were found to be predictive in prior data. And now we're looking at a COVID positive subset as described by some other condition. And if there's examples going on for that, but you could already see if I color by the amount of missingness, so the ones that are always observed tend to be higher values of, say, bilirubin, you're going to have to you probably get some biased inferences and biased estimates of prediction of something like, that's currently of interest, long COVID. But you may be able to make use of other variables like platelets and, given them the mixture that's more in the lower left to try to appeal to missing at random assumptions, right? So if you really suspect that this may be the case, now this is where I'm supposing we have full access to the data, right? And we know which ones are nearly always missing, we nearly always observed, nearly partially incomplete. But here, if you work with domain experts to suspect where these systematic missingness might come in, and where you may not have a random sample given your fully observed variables, you want to be able to have some domain expertise to say, what else do I have to bring in to try and appeal more closely to this missing at random assumption? The missing at random assumption that is, the missing information, those pieces are missing as a random sample stratified given other observed variables. And so, basically, you want to go beyond the complete case-only analysis model, cover your eyes and hope for the best, cross your fingers. It's a very strong restrictive assumption, not going to be generalizable, and at peer review, you're going to be caught on it. So the best thing to do is have a primary analysis that adopts missing at random with additional sensitivity analyses that examine plausible departures from that, not missing at random. How, if those that have incomplete information, if they're different from those that do have complete information, how do they differ? And how strongly would they differ in a plausible range of differences? In the end, you're going to have some assumptions there, whether you explicitly make them, they're going to be there implicitly, depending on what your analysis is, and it's going to drive your findings. So you got to own up to it. So lastly, if not to get into the this very restrictive subset of missing at random, there's always lurking under the surface this missing not at random. If you stress test that and your findings are similar, it's going to hold up better not only in peer review, but you can have the confidence it's more likely to be generalizable. So besides knowing your primary estimate and target, you got to know your data, and it's less than ideal provenance, just the way Hythem outlined. You've got to then also get that domain expertise to stratify by observables to approximate missing at random, use auxiliary data wherever possible, like the metadata, the provenance in N3C and how it's built from our data partners, and then narrow that range with further domain expertise as needed. Involve those data liaisons and logic liaisons who help build that pipeline to bring the data to you and your own domain team, your project team, and then stress test those primary findings by having some plausible missing not at random departures from the missing at random methods. And here's a quick outline of that. That's a quick rundown. These slides will be available, and there's some link outs to resources that have been used in simpler situations like clinical data. But take again stock about how there's so much already done, how many wrinkles are already ironed out for us by the N3C infrastructure, like the privacy preserving record linkage that deduplicates information across individuals, across different distinct data partners, like the folks in the research triangle area, where you may have UNC data as well as Duke data coming in to the N3C. Those are tied up, and what might be missing from one would be represented in the other. The idea is, though, to make use of all the observed information you have at your hand and work from it there. So to go on to a commonly used method, multiple imputation, we can see how you get this assumption of what the missing bits are off of the observed, such as it would be for BMI. And that's where we'll go to, in this fourth bullet, my colleague, Elena Casiraghi. Elena, I can advance the slides for you.
Thank you very much, Ken. I think you guide the presentation, or should I share the screen?
I'll advance for you if it'd be quick.
Okay, so we had the problem of BMI missing, so we need to try to infer the missing values for BMI, and we decided to revert to multiple imputation. So the problem behind the difference behind the commonly used inference when we have complete data, is that, okay, when we have complete data, we simply run our statistical analysis to get a statistical estimate or to get any inference we want to get, and we get our final estimate. But can we go to the other slide? Okay, what happens when we want to use multiple imputation? So probably everybody of you knows the multiple imputation pipeline. You have an incomplete data set, you try to impute it several times, where the number of imputations depends on some math, which I won't introduce. I will introduce if someone of you wants to know about that, but you simply produce multiple imputations of your data. On each imputed data set, you apply your inference model and you get different estimates, which you then pull at the end to get a pooled estimate. So, which is the difference between the left and the right? On the left, you have an estimate that comes from observed data. So from the complete data, I go to any inference that you could get from the complete data. On the right side, you have a pooled estimate that you would actually want to understand, and you on that estimate you want to know if that's a reliable estimate. Next. Could you go on the next? Thank you. So how would you choose between different multiple imputation models? If you search in the literature, you see that there are so many models. So there are artificial intelligence-based models like MIS-Forest, MIS-Ranger. There are statistically based models, and there are simple models like K-9 imputation model. So quoting Van Buren, which is a person that wrote that book that is written at the end of at the bottom right of the slide, the goal of multiple imputation is to obtain statistically valid inferences from incomplete data. And the quality of the imputation method should therefore be evaluated with respect to this goal. So in practice, what does it mean? In practice, this means that if we want to test, if we want to obtain, if we want to understand which is the best imputation model among all the ones we find in the web, we find wherever in all the repositories we have access to, we should compare the result of a day imputation algorithm on the for what regards the estimates that the imputation algorithm allow us to obtain. So if we go to the next slide, we go back to our slide, and we would like to have a complete data set. We would like to have the same version of the data set with incomplete data so that we can run the inference model on the complete data and get our estimates on the left. We run our multiple imputation pipeline with different multiple imputation algorithms. We get several pooled estimates, one per algorithm, and then we simply compare the estimate I got with each imputation algorithm with the estimate I got from the complete data. But unfortunately, if we go on the next slide, the reality of the fact is that we don't have the complete data, but we start with incomplete data. So how can we get those estimates on the left? And if we go to the next slide, thanks God, the N3C provides us the solution, because in the N3C we have enough data. We have a dream coming true for the computer scientists. So we have enough data, enough medical data, so we can take the incomplete data. We can just consider the complete cases, and in this time we get a complete data set. So on the complete data set, we can run our inference model to get our estimates on the complete data set. So those estimates are certain because I got them from a complete data set. So if we go to the next slide, now, how do we test our different multiple imputation algorithms? Well, we can produce what's called an amputated data set. So we take our complete data set, we produce some missingness, so we synthetically generate some missing values that are not random, but they are produced in a way to produce a missingness pattern that is similar to the one we had in the incomplete data, the one on the top left, the one with the sad man, so the sad smile. So I won't go in detail through this because also this would require more slides and more time. But once I have my synthetically generated also called amputated data set. I can run my imputation pipeline. I can get my estimate for the model, for the imputation algorithm I'm testing in this model. And now I can go with comparison, and I can compare the estimate I got on the complete data set, so on the data set that just contained complete cases, to the estimate I got on the amputated data set. So I have a first means of comparison between the imputation algorithm and the estimate I got on the complete data set. So if you go on the next slide, at the end, what we just need to do is compare these estimates. So the estimates on the complete data and the one I got after applying my imputation algorithm. Next. Okay, so we put in practice this on a cohort from diabetic patients where we wanted to perform, to obtain some causal inference estimates. Actually, we were applying two logistic regression model, one on the hospitalization event and one on the invasive ventilation event, and one Cox survival model on the death event. We obtain on the top some estimate on the complete data set. So on the full data set, after we have removed the incomplete cases, so on the pink box, you see a very little sketch of the estimate of the forest plot we produced to represent our odds and log odds estimates. On the bottom, you see what we did. So we produced multiple amputated data sets, not just one, because if we use just one data set, amputated data set, then our model could be biased through that particular amputated data set. We chose some appropriate evaluation measure to compare our log odds to the others. And then we could compare different imputation algorithms. Each algorithm was evaluated by using the pipeline you see sketch. So if you go on the next, I didn't go through the evaluation measures because they are those that we chose according to the problem we had at hand and to the fact that we were producing estimates that were odds estimates. And so at the end of the data set, the comparison we can obtain are in the next slide. And so here, this is just to show you that we could compare different models and we could assess different models, understand that misranger seems to be, misranger with no predictive mean matching seems to be the best estimator, the most reliable estimator, but also, for example, a version of mice can produce good results. We could understand the dependency of the different steps of the results we got from the different parameter setting of the different imputation algorithms. So we have realized the full pipeline to compare different imputation algorithms on your problem. So on each problem, we can apply this pipeline to choose which model is the best for our problem. And that's it. You can go to the next and proceed with Harold. This is just a wind tail loss to show you better the results if anyone is interested. So I can leave. This is another explanation I inserted if someone wants to have a good read on the way to choose the number of multiple imputations. And I leave my floor to Harold.
Okay, thank you, Elena. And let me share my screen. Please stop screening. Thank you. I know we're a little tight for time here, so I'm going to kind of zip through. You've heard a lot of details, and I want to show where we are in a project to kind of make this work easier. And reporting on work that I started about last summer and that Katie Bradwell and I have been working on implementing. So basically the problem that we're dealing with, the enclave is a complicated place, as we have made very clear this past hour. It's not always clear where to go. There are a lot of pieces. The analysis is complicated, and there is really no great lab notebook feature within the enclave. So our solution to this is to build a kind of computable protocol, an object in the enclave. There are 3 levels of protocols. There's the protocol design, basically the IRB version. There's a protocol in development as you instantiate your intended criteria. And there's a protocol that actually happened and which is really used for the publication. So as an example, this is pre-alpha. I just want to give you, we've spent a lot of time and just coming up with the bare bones to show where we're going. And I think we're ready to show that. So you can create a new protocol. There's administrative information. There's URLs and URIs that it gives you. You can edit a protocol where you can design sections, or so a protocol has different phases, and within each phase there are sections and examples of different types of sections. These are properties. These are all things that we want to be able to connect between the protocol as it's being worked out and all the resources that you're hearing about, whether it's be the templates or the best practices and so forth. And then as realized, you link to the final figures and tables, the variable dictionary. And if we can use certain ontologies and keywords and tags, we might be, the goal is to link the resources that one has specified through this whole method, not only to decision support using this, right now there are about 600 terms in here, but also, you may not have noticed, this methodology checklist, Ken showed you a couple of them. The idea is that as you build the information in the protocol, as you are working, you are perforce filling in the methodology checklist, and the reverse. At any point in time, you can go back and see what you've missed. So that's this basically a foretaste of where we're going. Folks, back to you.
Great. Well, those were really some great talks. So we have sort of a strategic issue here. So Peter and Justin, how long is your talk? I'm wondering, would it make sense to schedule it maybe for the next time or two, or do you have that flexibility? We have only 6 minutes left.
Yeah, it's up to all of you. Peter, what do you think? We could probably go next time. Or
Yeah.
But that makes sense because it's I'm sure has lots of content. Okay.
I mean, the summary was it's incredibly cool, but that's probably all we can say in the time available.
Okay, so let's iterate. Usman, are you on? Okay, so we'll iterate with Usman and sort of work out the scheduling. Okay, so that gives us no, thank you. I appreciate that because I want to make sure that we actually give some time to this. Title for next week, incredibly cool. All right. Okay, so we have 5 minutes then for discussion. So any questions, thoughts?
First of all, you should recognize, Elena, what, it's like midnight for you?
Yes.
Yes. So
my question would be, could you please move it a bit before this meeting?
So I wanted to get extra credit for the devotion that she's showing in
yes.
joining us. That was my first question.
Oh, okay, that's right. Yeah. So you're. Oh, okay. I'm sorry, I didn't parse it right because you have such a nice sunny background, it's not intuitive that you're in Milan. It's like a little late. Well, no, I appreciate that's really fantastic. And is there in terms of, and speaking of being, you know, midnight in Milan, I mean, these are all very generalizable methods. Do you happen to have, you happen to be working with European EU groups looking at these sorts of things, and are these methods, are you using these methods?
Well, we mainly work with other European groups. I actually work on images, on immunohistochemical images, where we have on the images, luckily we don't have holes, but then we pull the result on the images with medical data, with genetic data and medical data from cancer patients. So in that case, we have to apply multiple imputations. Yes.
I see. What sort of images?
They are immunohistochemical images because we work on the immunotherapy. So we are interested in studying the immunological response to cancers with the task of precision medicine, of developing new targeted chemotherapies for patients. So we work with the sarcoma group, the French sarcoma group, the Spanish sarcoma group, and of course the Italian tumor hospital. And then of course with all the hospitals in Italy and in the Lombardy area mainly. So my interest for BMI became deeper and deeper during COVID, when they called me to ask for a means to provide them some rules for understanding who were the patients at higher risk for dying. Because in the beginning in Milan, it was I think they lived the crazy times. They are my friends, and still when we talk about these times, all the clinicians, you can see a cloud in their eyes. So I remember that evening they called me asking, please apply some of your computers. They call it like that. Can you do a computer that can predict the severity of patients? Because we don't know how to handle them. Because they were saying that they were coming into the ER, that were walking, after a few minutes they were dying. Other ones, they were coming in lying, and after 2 days they were going home. So they couldn't understand how to treat it. So when they asked to apply some explainable artificial intelligence tools, and they gave us a lot of data with missing values, I understood that we couldn't just do as Ken brought, just let's impute and cross our fingers. We had to do things in a thorough way, and we had to experiment it in a thorough way, and we had to be sure that what we gave them, since it was to be applied in the clinical practice, wouldn't have killed the people. So I started investigating on BMI to understand, not on BMI, but on multiple imputation, to be sure that what I was doing was actually effective. So this is where everything started, and then 2 years have gone. So, and I have seen in these 2 years a lot of multiple imputation papers. So it seems that a lot of people they are having my same problem. Also because in the past few years, we have seen the explosion of digital health records and images, because during COVID, we have seen a lot of images. So it was a sort of a good advertisement for us, but on the other side, it was a bad period.
Yeah, no, thank you. And yeah, New York was like that too. And it's it is interesting because a lot of people in the N3C, or many people in the N3C, are, come from a sort of cancer cancer research area. Obviously, most people weren't working in infectious disease at the beginning of this. So, all right, well, we're over time. These were really extremely valuable talks. Really appreciate these great talks, and thanks for staying up so late. And look forward to seeing everybody next week. Thanks.
Ciao.
Ciao.